All right. Good morning, y'all. Welcome to part 2 of our Exam 1 review session. Real quick, in the Exam 1 review session materials on our course page, it's under the Unit 1 resources folder. Don't forget about these Kahoot review games. Only, I think it's 50 people can play before it just like maxes out. So if you ever go to play one of these Kahoot games, and it doesn't let you, just send me a message because I don't check them throughout the day. But let me know if they ever don't work. All right, so again, we're going to be using this Exam 1 review worksheet that's right here. So please have that handy. All right. You can't get into what? Oh, why is that one not available? Please hold. Let me check the other ones while we're sitting here. I made all of these available last night. Blackboard's been having, yeah, that one's available for that
It just
class.
kept taking me to the final page, like the award page or whatever it's called, the podiums. And then if I went back to the main Kahoot page and put in the game pin, it wouldn't work either.
Then let me see, which one? I guess it should work for any class. Do you remember, was it 1 or 2? Or
I
both?
tried both,
Ugh.
and on different days.
That's because it's been played more than 50 times. Okay, let me make a note. I will update those. Yeah, anytime y'all see something like that, please let me know. Because it turned like, so say Joe wanted to do the Kahoot and he wanted to do it 5 times, he can. And so sometimes when people want to play them over and over again, which is totally cool, we go through it really fast. So hold on 1 second. I got to find paper. I packed my office yesterday. Hold on. All right, so I will get those. I will get that reset. I'll probably do it while y'all start on this. So let me get y'all started on what we're going to do, and then I'll update those Kahoots. So thank y'all for letting me know. Okay, so let's start. Let's see, which monitor is this going to show up on? Okay, so today we're going to focus on things we didn't cover yesterday, but I do want this to be kind of a comprehensive review since it's our last time together before exams. So I threw in a couple of stuff from yesterday's review session, but in a little bit different of a format. So go ahead. I'm going to take about 3 minutes and update those Kahoots real quick. But go ahead and tackle questions 1 through 10. And if it's false, make the statements true. And as always, y'all, please try not to use your notes right now unless you get through all 10 and you're waiting for me to come back. But try and just use your brain because on your exam, you won't have your notes. All right, I'll be back in 3 minutes. For those of you that have been here, ignore my voice. For those of you that may have just joined, we're working on 1 through 10 right now. And I'm going to be back in 3 minutes. I'm going to go update the Kahoots. So be working on those. All right, take about 15, 20 more seconds. All right, so go ahead and finish up whichever one you're working on. And in the chat, we're going to put T if you think it's true, or F if you think it's false. And here's what we're gonna do. Don't push it yet, but I want you to put whatever you got for number 1 into the chat, but don't push enter, okay? And then I'm gonna say 1, 2, 3, go, and that's when you're gonna push enter, because I want them kind of all at once. And it just kind of helps people feel more comfortable with it, so they don't feel like anybody's judging them, which nobody is. The point of this is to be learning. So if you get it right, great. If you don't, great. That just means you're learning something. So please don't be shy. It kind of also helps me to see if I need to elaborate on something, or if it's like, okay, cool, got it. All right, so for number 1, go and put T or F in the chat. While I babble sometimes, if you want to correct the sentence, if you think it's false, you can also put that in too. All right, so go ahead and put T or F for number 1. And then again, I'm going to say 1, 2, 3, go. And on go is when you're going to push enter. Let's give this a shot. 1, 2, 3, go. Yay, look at y'all. Excellent, good job, y'all. So yes, so remember, conjugation is a form of horizontal gene transfer, and it is a true statement. The pilus does, remember, apoptotic fishing line, grabs a recipient cell that does not have the F plasmid, okay? So the donor cell has it, the recipient cell does not. The pilus is used to bring the recipient close by. It then forms a mating bridge. It then shares one strand of the plasmid, and then each of them will copy and make a second strand. So at the end of the day, both cells have a double-stranded DNA plasmid. So real quick, the reason I actually put this one in there, 1, to test you on conjugation, but 2, to remind you of a plasmid. Okay, remember plasmids. Emily, say I think it's fun too. I love it. Okay, plasmids, remember, it's too early, y'all. I got up too early. Okay, let's try that again. Plasmids are extra chromosomal. That means they are not part of the chromosome. They are not necessary for a cell to survive. But they offer some sort of bonus feature, and that's going to vary from plasmid to plasmid. Some code for toxins, which obviously help pathogens out. Some code for antibiotic resistance, also helps bacteria out. So they're helpful, but not essential, okay? All right, number 2, the codon, and y'all read these slow. A codon has the sequence CAG. The anticodon that matches it, that goes with it, I should say, has the sequence GTC. In the chat, put T or F, but don't push enter. If you think it's false, feel free to correct it. All right, 1, 2, 3, go. All right, let's see here. Oh, it goes pretty quick. All right. Ooh, Taylor even bolded. All right, so yeah, so this is why you got to look at it kind of slow. No, Taylor, perfectly fine. I'm that way too. Okay, so let's walk through this, y'all. So remember, what is a codon? It's like, stop there. What's a codon? We talked about this yesterday. Can anybody tell me? 3 nucleotides on messenger RNA. So right off the bat, our codon is fine. It's 3 nucleotides. RNA does have CAG. So far we're okay. When you get to anticodon, remember, anti means opposite. So that means it has to be complementary, okay? So if you look at it, GTC, that T cannot be there because we're talking about RNA. Codons are on messenger RNA, and T codons are on transfer RNA. So remember, there's no T in RNA. So that's why that one's going to be false. So the anticodon should be GUC because that's complementary to the CAG. Remember, G pairs with C, U pairs with A, C pairs with G. So that's how we did that. Good job. All right, number 3, put T or F in the chat. 1, 2, 3, go. Good, excellent. A pathogen does cause disease, and we're going to use that word so many times this semester. So you got to make sure that's locked in your brain. All right, put T or F for number 4. I'm going to count you down in just a second. The formation of biofilms is one way that microbes benefit humans. 1, 2, 3, go. Good. Yeah, it's so much easier to put false ones on reviews. On the exam, I tend to try to give a nice mix. Good. So biofilms are bad. They're harmful. Okay, well, they're bad for us. So yes, as Taylor mentions, remember, biofilm is a community of microbes. All different types of microbes can get together, but they grow up, kind of up and out. So if a biofilm starts in a heart valve or a catheter or something, it grows up and out and eventually fills it. It blocks it. Okay, so obviously we don't want our heart valves to be blocked or catheters. So biofilms are negative. Okay, so some ways that they benefit us, remember bioremediation. Remember, that's where microbes are used to remedy an issue. So the one I talk about in the videos is our oil spills. There just so happens to be microbes that help, they literally eat or metabolize oil. And they just so happen to not throw the ecosystem out of whack. So there's a lot that goes into finding microbes that can be used for things like that, but it just so happens with oil spills that is one. Let me see, a pathogen, oh, is which one? Jordan, can you elaborate on that for me? Is what specific to prions? Then I'm gonna kind of, oh, no, so biofilms, oftentimes it's bacteria, actually, because remember, prions are a colony of living organisms. And so prions aren't alive. Remember, they're just rogue proteins. So they aren't biofilms. But prions do aggregate together in brain tissue. So we kind of use different terms. Biofilm is going to be for living things that are constantly growing and kind of making our life more difficult. But prions do get together in little groups and insert themselves into brain tissue. Good question. All right, so bioremediation is one benefit. We talked about recycling nutrients, and I don't know if I took this out or not, but wastewater treatment, we actually use a lot of bacteria to kind of break down some things in dirty water. And pest control, some bacteria naturally release things that keep pests away. And so then remember, they're dangerous. Microbes can be dangerous because they're pathogens. They cause our food and stuff to go bad, biofilms, things like that. All right, so let's kind of switch gears now to parts, the cells conversation. So number 5, go ahead and put T or F in the chat. Cilia are hair-like projections that cover some eukaryotic cells. 1, 2, 3, go. Really, I love yelling go. I think it's fun. Hey, excellent, y'all. And there was a lot here. So as y'all probably know by now, true false isn't new to any of you. You really need to take it one word at a time, right? So cilia are hair-like projections. That is true. They're little, remember, cilia are small and they cover the organism completely. So then that's what I say next, that covers some eukaryotes. And that's going to be true. Now, some of you may have already put T or F in the chat, but feel free to delete it and answer this question. Prokaryotes, bacteria specifically, also have little hair-like projections that can completely cover them, but they have a different name. Does anybody remember what that's called? And don't worry about spelling. Not quite. It does have an F. It starts with an F. Flagella are long, whip-like, tail-like structures. Yeah, there we go, fimbriae. So remember, fimbriae are accessory structures, okay? And they're little hair-like projections that help some bacteria attach to surfaces. So when we get into Unit 3 and we talk about how pathogens actually get into the body and make us sick, a lot of pathogens have fimbriae, and that's how they kind of adhere to our cells and cause, you know, chaos in our body. So yeah, fimbriae are little hairs. Flagella are those long tail-like structures. But y'all are definitely on track with the F's. Okay. All right, moving on to number 6. A capsule is made up of sugar and helps pathogens evade our immune system. True or false? 1, 2, 3, go. Good. Yes, this one is true. And yes, Jordan, absolutely. So glycocalyx is like the formal word for a sugary layer. The capsule is when we're being more specific. It's very tightly attached to the cell. There are structures that are sugary, but they're loosely attached. And y'all don't have to worry about that for this class. But yes, that's the glycocalyx. And it's a really good thing to remember that, Jordan, because glyco means carb or sugar. So if you remember that the capsule is also called glycocalyx, you're going to go, oh, it's a sugary layer. And that should help you go, okay, sugar can be sticky. Okay, our immune cells get kind of stuck. They can't grab onto them to get rid of them easy. So that's one reason they evade our immune system for a little while. Does anybody remember what is the science word for like a weapon that the bacteria use to make us sick? The capsule is an example of this word. Does anybody remember? Technically, it's 2 words. Yes, good job. Awesome. Yes, virulence factor. So this is going to be really important as we get through unit 3 and beyond. Virulence factors are structures or molecules like toxins that bacteria use to kind of wage war against us. And so good job. Y'all want to get all these terms in your head. I know it's a lot. You're literally learning another language in this class. So excellent. So yes, this statement is true. Capsule is a sugary layer. And if we want to be more specific, it helps evade our immune system because it evades phagocytosis. And we're going to talk a lot about that when we get to our immune response. All right. Oh, it's already 10:20. Okay, I'm going to quit trying to babble too much. Number 7. Reddy attempted to prove spontaneous generation by boiling broth in uncorked flasks. All right, on 3, 1, 2, hey, 1, 2, 3, go. Guys, I didn't get my go. No, I'm just kidding. No problem. Okay, yeah, so this one is incorrect. And so here we go. Let's see. Good. Yeah, so really the easiest fix to this is changing ready to Needham. Needham was the one that this statement is true to. So ready was the uncooked meat in the jars, and he was trying to disprove spontaneous generation. Reddy is really the one who got the ball rolling in trying to discredit it. And so I remember Reddy because I think of, like, you know, if somebody's cooking dinner, like cooking a steak or cooking meat, you're like, hey, is it ready yet? And, you know, that's just what works for me. It's ready. In this case, it wasn't ready because it was raw meat. All right, whatever works for you. So we're going to talk more about these scientists in a minute, so I'm going to keep going. Number 8, the replication of some animal viruses is known as the lysogenic cycle. T or F? 1, 2, 3, go. All right, so yes, this one is false. So yeah, the incorrect part of this statement is this is not an animal virus thing. This is bacteriophage. So remember, bacteriophage are viruses that only infect bacteria, so they're not like directly a threat to us. You may have actually heard of phage therapy, where we use bacteriophage to deliver drugs or you know, treatment to like cancerous cells and other things. It's pretty fascinating. We'll try and talk about it more when we get to the diseases. But we can use that because bacteriophage don't hurt us. They're harmful to bacteria. So it's pretty snazzy. The lab that I worked in as an undergrad, that dude, super smart. He's actually one of the leaders in phage therapy. Pretty snazzy. And here I go digressing. It just comes naturally, y'all. All right, so remember, if you see lysogenic or lytic, those cycles are bacteriophage. Okay, we're going to walk through animal virus replication in a little bit, but lysogenic, lytic cycles, those are bacteriophage. All right, number 9, all viruses have genetic material and an envelope. True or false? 1, 2, 3, go. Taylor, you really capitalize on my babbling. I love it. All right, good. This one is false, okay? So there are 2 things that all viruses have. Some form of nucleic acid. We call it genetic material. For those of you that have me in person, I kind of rambled on this a little bit. I don't like calling it genetic material, although I typically do because it's what people tend to say. But because they're not alive, I don't like using that term the same as we do for living things, where it is our genetic information. But because nucleic acids do kind of function similar in viruses as they do in us, it's okay to use it. It just makes me a little eh. But so yeah, they all have some sort of nucleic acid and they have protein. Remember, that nucleic acid needs to be protected, just like it does in living things. And so remember, the capsid is that protein coat that surrounds and protects the nucleic acid, okay? All viruses also have spike proteins, but those are also protein, okay? So nucleic acid and protein are the 2 things that all viruses have. For very structurally simple viruses, the capsid proteins can serve as the spike proteins. For more complex ones, like you saw in the content videos, sometimes the spike is a very different protein from the capsid proteins. All right, so remember, yes, envelopes are what we call accessory structures. Not all of them have it, but remember, they steal it from the host. The envelope is actually host plasma membrane. And so why is that beneficial for a virus? Why would they want to steal part of the host plasma membrane? I think I talked about this a little bit in the video. There we go. Yes. Yep. It's a form of protecting themselves specifically because, exactly, Shelby, they're protecting themselves by trying to be camouflaged, right? They're trying to blend in within the host. And so they're disguising themselves as a host cell, or they're trying to, which is pretty clever, right? But for enveloped viruses, it may delay the inevitable, but it's those spike proteins, because enveloped viruses tend to have the big spike proteins that stick out, and that's eventually what our immune system will detect. Good job, y'all. Okay. That's the end of our true false. All right, good. Now, short answer, I'm just going to give y'all, just take 1 minute to just kind of jot down some things, and we'll talk through 11 through 13. So I'm going to be quiet for 1 minute, if you can imagine. 1 minute while you just kind of jot down some thoughts for 11 through 13. All right, I think that was about a minute. So 3 ways viruses are diverse. So hopefully you've all either listened to me in person about ramble on about viruses or the content videos. And there are 3 big ways that viruses tend to differ from one another. And because the big point I try to drive home is comparing one virus to another is just not a good idea, okay? Because viruses just don't behave similarly to other viruses, which is unusual, right? It's very hard for the general public to understand because most cats behave like other cats. Most monkeys act like other monkeys, right? Same thing with humans. And so it's logical to just assume all viruses must be the same, right? But we know that they're not. So what are 3 ways, and you can just give me some keywords in the chat. I'm not going to count us down for these, but what are some different ways that viruses are diverse? Go ahead and put it in the chat when you're ready. You can throw out 1 thing, 3 things. Good, Garner. Yep. Protein shape. I'm going to modify what you wrote just a little bit. That protein coat is the capsid. So yeah, the shape or structure of the capsid specifically. Very good. Yep. Ooh, host range. Jordan, good. I didn't even think of that, but you're absolutely right. The host range, good. Spike proteins and host receptor, good. That's going to determine if they, yep, good, good, good. All right, Emerald, yes, genetic material is 1. So that's what I had in mind. And so this is why I love these reviews, because people put things I hadn't even considered. So I'm going to put nucleic acid. So remember, y'all, I'm going to be abbreviating on here. So some viruses have DNA, others have RNA, but never both. So some viruses have double-stranded DNA. That's how we abbreviate it. Some have single-stranded DNA, which is baffling to us. Some viruses have single-stranded RNA, which kind of makes sense. And some have double-stranded RNA, which again doesn't make sense to us. We don't have double-stranded RNA. So that's one way they're really diverse. What else, aside from what's already been said? Oh, we did have capsid. That was one of the ones I was thinking of. Capsid structure. And we can actually just be more vague and say structure in general. That goes with the capsid. That goes with, is it enveloped or not? Does it have, like, I'm going to call them fancy spike proteins, the ones that stick out, or is it just using the capsid? Right, lots of diversity in overall just the structure of the virus. What's the last way that they're different? We spent a bit of time in class talking about this. Same on the videos. Replication, right? Because the genetic material is going to dictate how they replicate. DNA viruses replicate differently than RNA viruses. Okay, so yeah, excellent, Jordan, exactly, how they replicate. And so that's what 12 and 13 are all about. So let me go ahead and put a little bit of notes here. So lytic and lysogenic. So again, these are for bacteriophage, okay? Viruses that only infect bacteria. So remember, for the lytic cycle, okay, the goal is to get into a host, replicate, and then get out of the host. They don't stick around, nothing like that, okay? And so that's kind of the goal there. Now remember, there are 5 steps. Can anybody tell me some of the steps for lytic replication? Good, Garner, yes. So first they need to attach. Then they are going to inject their DNA into the host. That step is called penetration. Okay, good. And then once that nucleic acid gets in, okay, so remember this, y'all, this is really important. Bacteriophage only insert their DNA into the host. Remember I said the bacteriophage have that creepy looking structure where it's got the little creepy tail fibers and things. All of that stays outside of the host, okay? Only the DNA gets in. This is why they can automatically move to biosynthesis, which means they're making all the things needed to create new viruses, okay? If they're going to make 100 new bacteriophage, they need 100 copies of that DNA. They need 100 copies of the capsid. So that's what biosynthesis is. It's making all that stuff, all the viral stuff. Once all of that has been made, they mature, which is basically they're spontaneously assembling. They get together and form the new bacteriophage, and then they exit, and they exit via lysis. Okay. So again, they get in, which that's the attach and penetrate part. They replicate, that's biosynthesis and maturation, and then they get out, and that's going to be lysis. Okay. Good job, y'all. So then the other way that a bacteriophage might replicate is lysogenic. So here they're going to get in, and then this is what I call integrate and chill. And then they're going to replicate and get out. So there's a lot of overlap between lytic and lysogenic. There's just one extra part to the lysogenic stage, okay? So I'm going to type it out as I talk about it. So they're going to attach as well, attach to the host bacteria. They're going to do penetration, where again, that viral DNA is going to get injected into the host, but this time, the viral DNA integrates into the bacterial DNA, forming a prophage, okay? That's the integrate part. Once it's integrated, they can chill. The bacteria divides like normal. It has no idea it's infected. So it just keeps dividing and dividing. Now all of those bacterial cells are carrying viral DNA, okay? And then eventually it gets stressed out. Poor bacteria have stressful lives. The viral DNA exits the chromosome, and then it's going to do biosynthesis. I can't type, but I'm not going to worry about it. Maturation, and eventually it gets out as well. Okay. So notice, y'all, people tend to think that these two cycles are so different from one another, and they're not. Okay. The difference is going to be, like I said, that integrate and chill idea. But they have all the same 5 steps as the lytic cycle. Any questions about bacteriophage or anything up to this point? Let me see, Jordan. Is it only a prophage when integrated? Yes. Yep. Nope. Prophage is the term for when phage DNA is integrated into host DNA. And then if you think about the retrovirus conversation, remember some animal viruses can do the same thing, but we call that a provirus when they're integrated. Phage tends to be a bacterial term when it infects bacteria. Virus is normally used when it infects animals. So prophage and provirus are essentially the same thing. Viral DNA into host DNA. Prophage is if the host is a bacteria. Provirus is if the host is an animal. Good question. All right, so now we're going to outline the steps of animal virus replication. And y'all, again, big picture, same thing. Get in, make more viruses, get out, right? Animal viruses tend to be a bit more sophisticated because our immune system's going to go bonkers. But I'm going to go ahead and walk through this. Feel free to, if you want to put anything in the chat, you can. So it's going to have the same thing. It's going to attach. Remember, that's where the host range comes in. A virus can only attach to a host if the viral spike proteins are complementary to the host receptor. Okay, so it's going to attach. Okay, and then it's going to enter. Notice this is not called penetration. Okay, because with the bacteriophage, they injected only the DNA into the host. That does not happen here. For animal viruses, that genetic material gets into the host protected, either by the capsid or by the envelope or whatever the virus has. Okay, so we don't call it penetration because it's 2 different things here. So some viruses enter by fusion, others enter by endocytosis, and there's pictures of both of those on the PowerPoint and the videos. But remember, no matter what, during this enter, or maybe it's called entry phase, point as it gets in, remember why this is important is the genetic material is not exposed, but it needs to be, because it can't take over the host cell until the genetic material is exposed. Because once the host cell detects DNA, it doesn't, or RNA, it doesn't know that it's viral. Okay, cells aren't that sophisticated, but cells are programmed to just naturally be like, oh, hey, is that DNA? I better replicate it. Oh, I better do transcription translation. It's just an automatic thing, okay? So that's why it's essential the viral genetic material be exposed. So the process of exposing animal virus genetic material is called uncoating, because that's where the virus is essentially falling apart to expose that genetic material. That's the big difference between the bacteriophage and animal virus replication cycles, okay? All right, once this happens, it's going to go through biosynthesis. It's going to do transcription and translation, make all those viral proteins. It'll do replication to make more genetic material, okay? Once it does that, they're going to spontaneously assemble, so maturation, and then the viruses are going to exit, okay? Naked viruses, and viruses that do not have an envelope are going to exit by essentially blowing up the cell. Lysis, they go all ninja and poke holes in the host and leave that way. If the virus has an envelope, then it's going to leave by budding. Any questions on animal viruses? So, good question, Jordan. Okay, so fusion, you're absolutely right. The membrane becomes continuous with the cell. I'm gonna rephrase that just a little bit. Only enveloped viruses can do this, because remember, the envelope is host cell membrane, right? So when it goes to infect another cell that also is host cell membrane, they can fuse together, because host membrane is host membrane, right? So for enveloped viruses, that envelope fuses and becomes continuous with the host plasma membrane. But when that happens, it's dumping in the capsid and the genetic material, okay? So it would still need to uncoat, because that genetic material is still protected by the capsid. So the capsid would need to fall apart. Yeah, great question. It's a very good question. All right, any other questions? Awesome, y'all are doing great. Okay, so I'm gonna get y'all back involved, a little less of me babbling. So let's go ahead and just do 14 through 16 for right now. And let me get this all on one page, because it drives me nuts if it's not. All right, so I'm gonna give you just a second, and then we're gonna do the same thing. I'm gonna have you put the answer to 14 in the chat. I'll count us 1, 2, 3, go, and you'll put it in there. And then we're gonna use these questions as a platform to talk about all the different answer choices. So if you've already got your answers for 14 through 16, start making notes. What would the question needed to have said for the other ones, a different answer choice to be correct? What do you need to know about the Golgi, the ER, the vacuole? Do you use this as kind of like a brain dump? All right. Number 14, in the chat, put which letter you think is correct, and then I'm going to count us down. 1, 2, 3, go. Excellent. The endoplasmic reticulum. So remember, y'all, biosynthesis means it's making things the cell needs to survive. So think about our macromolecules, proteins, carbs, lipids. The only thing it doesn't make is nucleic acid, right? Because that's going to be DNA replication. Okay, excellent. So the ER, remember, we have smooth and rough ER. Rough ER is covered in ribosomes, and we know ribosomes are protein-making machines. So the rough ER is where a lot of proteins are made. But there are ribosomes elsewhere inside the cell because every cell needs tons of proteins, tons of different proteins, lots of copies of each of those proteins. So there are ribosomes on the rough ER, but there's also ribosomes just hanging out elsewhere inside the cell. Sorry. The smooth ER is going to handle lipids and carbohydrates. All right. So can anybody tell me, what do vacuoles do? What's kind of their function? Exactly. They're storage compartments. They hold on to food, hold on to water. In our cells, they'll hold on to pathogens. Okay. It just holds stuff. Yep. Excellent. Good job, y'all. All right. The nucleus, its job is to protect the DNA, right? That's where the DNA is going to be made. So again, if we're even talking about a nucleus and Golgi and ER, we have to be talking about eukaryotes, right? And so because DNA is in the nucleus of eukaryotes, that's where that's where DNA replication will happen. It's also where transcription will happen. Okay, the Golgi. So the Golgi, its big job, it has two: modify and distribute. A lot of people think of the Golgi as like a post office, or even like a UPS center, I guess, or whichever distributor you prefer. But really, it just takes in all the stuff made in the ER. It makes sure it's good. It makes any changes if necessary, and then it ships it out to wherever that molecule needs to go in the cell. It distributes it. All right, and then the lysosome, which is my favorite organelle. The lysosome is, what does it do? Y'all tell me. Oh, sorry. Good. It's going to break things down. It digests things. So it's either going to break down food, or it could break down a pathogen. Its whole purpose is digestion. Okay. So if you've learned about phagocytosis, I think A&P talks about it. But the whole point of phagocytosis, right, is when our immune cells catch a pathogen and it needs to eliminate it. It chops it to bits. Well, the lysosome is a key part of that process because the lysosome is acidic. That's what's really unique about the lysosome, because in order for the lysosome to digest things, it has to have hydrolytic enzymes. I'm going to put that in the chat. It's a good thing because we're going to talk about phagocytosis so much. This is something that you just want to be familiar with. So hydrolytic enzymes are the little specialized proteins that chop things into pieces. But in order for those hydrolytic enzymes to work, they have to have an acidic environment. So the lysosome is the only really acidic environment in the cell. So that's where these little hydrolytic enzymes are going to work. Okay? So those are some important things to remember about the lysosome. Okay, excellent. Let's go to number 15. Y'all definitely make sure you know all about each organelle, just kind of like what do they do? So not on here, let's see, is what the chloroplast and the mitochondria. So make sure you know those two. All right, 15. Go and put your answer into the chat and I'll count us down in 1 second. 1, 2, 3, go. I guess, am I counting us up? Okay, which scientist supported, yeah, so we actually talked about this one earlier, Needham. So remember, let's see, actually there's only, I guess there's 2 others that were involved in the spontaneous generation debate. Needham was really the big supporter that we talked about. Spallanzani and Pasteur, along with Reddy, those 3. Hold on, my dog's about to go crazy. My mom just got here. Hold on. That was really stealthy, Mom. I didn't think Kaya was singing. Sorry, she's coming over to help me with my yard because my HOA is mad at me. Okay. Sorry about that. All right, so Reddy was the meat person. So he was trying to disprove spontaneous generation. Spallanzani was trying to shut down Needham's work. So their experiments looked really, really similar, right? They both boiled beef broth. But Needham, the mistake in his setup was that he let the beef broth cool before he corked it. So he did see bacterial growth. Spallanzani put the beef broth in the flask, corked it, boiled it, and therefore there was no growth. All right, so Pasteur did a lot of stuff in terms of the spontaneous generation debate. He's the one who finally completely disproved it. Nobody else wanted to argue him. Pasteur was kind of a beast. Remember, so he did an experiment similar to both Needham and Spallanzani, but his key to success was he used S-necked flasks, because the big problem people were having was they were saying, well, there's no oxygen in your system, so of course nothing can grow. And so Pasteur, his S-necked flasks were open at one end. So air could get in, technically microbes could get in, but they just couldn't access the actual broth because of the S shape. So Pasteur was the one to finally shut it down. And then the other thing Pasteur did was fermentation, which we'll talk more about next unit, and pasteurization, which we'll also talk about next unit. But the one thing he did not win was the race between him and Koch to provide evidence for germ theory. So remember, they were both trying to prove that specific microbes cause a specific disease, okay? And Koch won because of Hesse. Remember, she was a cook, and so she was the one who suggested they use agar in their media to grow things, and so that was the missing piece. And so Koch developed his 4 postulates, which are basically guides to saying, yes, this one bacteria causes this one disease, and here's the proof you need. So that's what the postulates were. Kind of, you need to show this, 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 and this, and you can say that this microbe causes this disease. All right, any question about these scientists? Make sure to review all of them. Okay? You probably won't be surprised when you see something about them on your exam. All right, number 16, we're almost done. We're gonna be right on time. All right, which type of microbe is eukaryotic, lacks a cell wall, and is characterized by its motility? Put your answer in the chat. I'm gonna count us up. 1, 2, 3, go. The correct answer for this one is gonna be B. So let's talk about why. So notice eukaryotic. Okay, that's your first piece of information. So that's going to eliminate bacteria and the archaea, because those are both prokaryotic. Okay. Remember, archaea live in extreme environments, so they're not really a concern for us. Bacteria is what we talk about most in this class. So the remaining 3 on here are eukaryotic. Both algae and protozoans, they're what make up our protist category. I saw some people in my online class we're asking about protists because that's what's mentioned in the lecture notes. Protists are divided into two groups: the plant-like algae and the animal-like protozoans. But algae, hold on. Oh, how did I zoom in like that? Oh, what did I do? Okay, sorry, y'all. Okay, so algae do have a cell wall. Why is it showing up weird on mine? Okay, well, algae have a cell wall made of cellulose. Helminths, they actually, that's a good point. They don't, because they're animals, animal cells don't have a cell wall. But even though helminths do move, they're not characterized based on how they move. They all kind of move in a similar fashion. Helminths are actually, and you'll learn about this in Unit 5, they're characterized by the scolex, the head. A lot of helminths have like hooks or suckers or things that help them attach to our intestines. So the head region is one way we do that. But protozoans we differentiate the different protozoans based on, do they have pseudopods? Do they have flagella? Do they have cilia? Do they have nothing? So Taylor, that's a good point. I can see why B or D would have been what you narrowed it down to. But in this case, B is going to be the correct answer. All right, y'all, is it showing up like really zoomed in on what y'all are seeing? Because it's not zoomed in real life on my screen. Okay. Okay, interesting. Well, I'm just going to keep going as though it looks fine because we got 6 minutes. All right, so 17. Joey went to bed Sunday night feeling healthy. He woke up the next day with a stuffy nose and a cough. After 2 weeks, all the symptoms were gone. How would you describe this viral infection? Oh, sorry. 1, 2, 3, go. Good, yes. So remember, acute comes on quick and goes away quick for a viral infection. Persistent is where it lasts for months or years. Technically, it can last a lifetime, but not normally. Mono is a great example of a persistent infection. And then latent is going to be where it is with you for life. These are things like herpes, HIV, where those proviruses occur. The viral DNA gets integrated into the host DNA, and so it is with you forever. It cannot be eliminated. And so remember, with latent viral infections, there's outbreaks. So when somebody gets a cold sore, that's an outbreak, okay? And then it clears up and they're not contagious anymore. So latent infections have moments of outbreaks, or reactivation is the science term that y'all will learn. Those outbreaks are caused by viral reactivation. All right, take a look at 18. Review from yesterday. What is the start site of translation? 1, 2, 3, go. Yes, good. So remember, translation is starting with RNA and making protein, okay? So we're starting with RNA. Remember, that's what a codon is. It's a set of 3 RNA nucleotides. Origin of replication, that's DNA replication. Promoter, transcription. We didn't even talk about this. It's also transcription. You don't need to know that. Start codon, remember that is going to be AUG. Okay, so the answer is D. All right. 19 is going to need the codon wheel. If you have that handy, go ahead. I'm going to try to pull it up for you. I know we are running out of time. Where's my codon wheel? Why isn't it popping up? So remember the 4 different types, excuse me, 4 different types of mutations. I'm going to go ahead and so you guys will see the codon wheel. It's embedded in the test. So you don't need to bring your own or anything. You shouldn't bring anything with you for your test. And it'll be the one you're used to seeing. Just for the sake of time, I'm just going to use this one right now. So remember, to figure this out, okay, the piece of DNA should have been ACG. So I'm going to have to take this out. So ACG is the DNA. So the RNA it would make is UGC, right? Complementary, think of base pairing. Use the codon wheel to figure out what amino acid that should be. So UGC is going to be the amino acid cysteine. That's what should have happened, okay? Now, DNA polymerase makes a mistake, and instead, the DNA is AGG. Okay, so that second nucleotide is wrong. So think about how that affects the RNA. And then now look at what that does for the amino acid. So UCC is serine. So which type of mutation is this? Go ahead and just put it in the chat. I keep forgetting to count us down. Which type of mutation is this? Sorry, I'm distracted by my own typing. This one is actually going to be, it's a missense. Yep, because it's a mistake. Okay? Because if it should have been cysteine and it was cysteine, that would be silent. But because it's a different amino acid, it's a mistake. So that's a missense. Had this become a stop codon, it would have been a nonsense mutation. All right, let's go through these last two real quick. Which is not a key player in DNA replication? So which of these is the odd man out? Go ahead and put it in the chat when you got it. I'm not going to count this down. Good. Good. Transfer RNA. What process is transfer RNA used in? Good. Translation, because it's involved in making proteins. All right. Last but not least, which enzyme binds the promoter? So ask yourself, the promoter is the start site for what? What do we got? B. Yep, exactly. The promoter is the start site for transcription. That's where we start with DNA and make RNA. And so RNA polymerase is responsible for that. Excellent. Great job, y'all. We are right on time. If you have any questions, please feel free to put them in the chat. I'm going to go ahead and stop the recording so Blackboard can